Hey guys, welcome to Medifaction. So today we're going to learn about the physiology of nephron. So what is a nephron? A nephron is nothing but the microscopic structural and functional unit of the kidney. So in order to learn more, let's go in detail to learn more about nephron. So in this video, I'm going to enumerate an introduction about the nephron and followed by the renal corpuscle, in which we will discuss about the situation, classification of nephron, structure of nephron. Also, we are going to talk about the tubular portion of nephron, in which we will define the proximal convoluted tubule, loop of Henle, distal convoluted tubule, and also we are going to talk about the collecting duct, and at last we will end this session with passage of urine. Let's begin. Introduction. Nephron is defined as the structural and functional unit of kidney. Each kidney consists of 1 to 1.3 millions of nephrons. The number of nephrons starts decreasing after about 45 to 50 years of age at the rate of 0.8% to 1% every year. Each nephron is formed by two parts: a blind end called renal corpuscle or Malpighian corpuscle, and a tubular portion called renal tubule. So this is what a nephron looks like. Let's move to detail. So on dividing a nephron, there is two parts, that is a renal corpuscle and a tubular portion, as I mentioned earlier. So this renal corpuscle can be again divided into a glomerulus and a Bowman capsule. The tubular portion can be divided into a proximal convoluted tubule, loop of Henle, and a distal convoluted tubule. The loop of Henle can be again divided into three parts, which are the descending limb, the hairpin bend, and the ascending limb. Descending limb here can be again classified into thick descending segment and thin descending segment. Likewise, the ascending limb also can be categorized as thin ascending segment and thick ascending segment. Renal corpuscle. Renal corpuscle or Malpighian corpuscle is a spheroidal and slightly flattened structure with a diameter of about 200 microns. Function of the renal corpuscle is the filtration of blood, which forms the first phase of urine formation. Situation of renal corpuscle and types of nephron. Renal corpuscle is situated in the cortex of the kidney, either near the periphery or near the medulla. Classification of nephrons. Based on the situation of renal corpuscle, the nephrons are classified into two types. Number one is the cortical nephrons or superficial nephrons. Nephrons having the corpuscle in outer cortex of the kidney near the periphery. In human kidneys, 85% nephrons are cortical nephrons. Number two is juxtamedullary nephrons. These are nephrons having the corpuscles in the inner cortex near medulla or corticomedullary junction. Structure of renal corpuscle. Renal corpuscle is formed by two portions. Number one is the glomerulus. Here you can see this is the glomerulus. Number two is the Bowman's capsule. This part which encloses the glomerulus is known as the Bowman's capsule. So let's move to detail. Glomerulus is a tuft of capillaries enclosed by Bowman capsule. It consists of glomerular capillaries interposed between afferent arteriole on one end and efferent arteriole on the other end. Thus, the vascular system in the glomerulus is purely arterial. Glomerular capillaries arise from the afferent arteriole. After entering the Bowman capsule, the afferent arteriole divides into 4 or 5 large capillaries. Each large capillary subdivides into many small capillaries. These small capillaries are arranged in the irregular loops and form anastomosis. All the smaller capillaries finally reunite to form the efferent arteriole, which leaves the Bowman's capsule. Diameter of the efferent arteriole is less than that of the afferent arteriole. The difference in diameter has got functional significance also. Functional histology. Glomerular capillaries are made up of single layer of endothelial cells which are attached to the basement membrane. Endothelium has many pores called fenestrae or filtration pores. Diameter of each pore is 0.1 micron. Presence of the fenestra is the evidence of the filtration function of the glomerulus. Bowman capsule. It is a capsular structure which encloses the glomerulus. It is formed by two layers. Number one is the inner visceral layer. Number two is the outer parietal layer. Visceral layer covers the glomerular capillaries. Here we can see the Bowman capsule. It is continued as the parietal layer at the visceral pole. Parietal layer is continued with the wall of the tubular portion of nephron. The cleft-like space between the visceral and parietal layer is continued as the lumen of the tubular portion. Functional anatomy of Bowman capsule resembles a funnel with filter paper. Diameter of Bowman capsule is 200 microns. Functional histology. Both the layers of Bowman capsule are composed of a single layer of flattened epithelial cells resting on a basement membrane. Basement membrane of the visceral layer fuses with the basement membrane of the glomerular capillaries on which the capillary endothelial cells are arranged. Thus, the basement membranes, which are fused together, form a separation between the glomerular capillary endothelium and the epithelium of visceral layer of Bowman capsule. Epithelial cells of the visceral layer fuse with the basement membrane, but the fusion is not complete. Each cell is connected with basement membrane by cytoplasmic extensions of epithelial cells called pedicles or feet. These pedicles are arranged in the interdigitating manner, leaving small cleft-like spaces in between. The cleft-like spaces is called slit pore. Epithelial cells with pedicles are called podocytes. Tubular portion of nephron. Tubular portion of nephron is the continuation of Bowman's capsule. It is made up of three parts. Number one is the proximal convoluted tubule. Number two is loop of Henle. Number three is distal convoluted tubule. Now let's go for the detail. Proximal convoluted tubule. Proximal convoluted tubule is the coil portion arising from the Bowman's capsule. It is situated in the cortex. It is continued as descending limb of loop of Henle. Length of proximal convoluted tubule is 14 mm and the diameter is 55 microns. Proximal convoluted tubule is continued as loop of Henle. Functional histology. Proximal convoluted tubule is formed by single layer of cuboidal epithelial cells. Characteristic feature of these cells is the presence of hair-like projections directed towards the lumen of the tubule. Presence of these projections, the epithelial cells are called brush-bordered cells. Loop of Henle. It consists of a descending limb, hairpin bend, and an ascending limb. So this right here is the descending limb, which is the continuation of proximal convoluted tubule. So the descending limb of loop of Henle is made up of two segments, thick descending segment and thin descending segment. Let's talk about thick descending segment. Thick descending segment is the direct continuation of proximal convoluted tubule. It descends down into medulla. It has a length of 6 mm and a diameter of 55 microns. It is formed by brush-bordered cuboidal epithelial cells. thin descending segment. Thick descending segment is continued as thin descending segment right here. It is formed by flattened epithelial cells without brush border and is continued as hairpin bend of the loop. This right here is the hairpin bend of the loop. Hairpin bend. These are formed by flattened epithelial cells without brush border and is continued as the ascending limb of loop of Henle. This is the ascending limb of loop of Henle. Ascending limb. Ascending limb or segment of loop of Henle has two parts. That is a thin ascending segment and a thick ascending segment. Let's talk about the thin ascending segment first. Thin ascending segment is the continuation of hairpin bend right here. It is also lined by flattened epithelial cells without brush border. Total length of thin descending segment, hairpin bend and thin ascending segment of loop of Henle 10 mm to 15 mm and the diameter is 15 micron. Thin ascending segment is continued as thick ascending segment. Here we can appreciate that. Now let's talk about the thick ascending segment. Thick ascending segment is about 9 mm long and the diameter of 30 micron. Thick ascending segment is lined by cuboidal epithelial cells without brush border. The terminal portion of thick ascending segment, which runs between the afferent and efferent arterioles of the same nephrons, form the macula densa. Macula densa is the part of juxtaglomerular apparatus. Thick ascending segment ascends to the cortex and continued as distal convoluted tubule. Length and extent of loop of Henle. Length and extent of loop on Henle varies in different nephrons. In cortical nephrons, it is short and the hairpin bend penetrates only up to outer medulla. In juxtamedullary nephrons, this is long and the hairpin bend extends deep into the inner medulla. In some nephrons, it even runs up to the papilla. Distal convoluted tubule. Here you can appreciate the distal convoluted tubule. It is a continuation of thick ascending segment right here and occupies the cortex of the kidney. It is continued as the collecting duct, that is here. The length of the distal convoluted tubule is 14.5 to 15 mm. It has a diameter of 22 to 50 microns. Functional histology. Distal convoluted tubule is lined by single layer of cuboidal epithelial cells without brush border. Epithelial cells in distal convoluted tubule are called intercalated cells or eye cells. The collecting duct. Distal convoluted tubule has the initial or arched collecting duct which is in cortex. This is the collecting duct. The lower part of the collecting duct lies in medulla. 7 to 10 initial collecting ducts unite to form the straight collecting duct which passes through medulla. Length of the collecting duct is 20 to 22 mm and its diameter varies between 40 and 200 microns. Collecting duct is formed by cuboidal or columnar epithelial cells. Functional histology. Collecting duct is formed by two types of epithelial cells. Number 1 is principal or P cells. Number 2 is intercalated or I cells. These two types of cells have some functional significance also. Passage of urine. At the inner zone of medulla, the straight collecting ducts from each medullary pyramid unite to form papillary ducts or ducts of Bellini, which open into a V-shaped area called papilla. Urine from each medullary pyramid is collected in the papilla. From here, it is drained into the minor calyx. 3 or 4 minor calyces unite to form one major calyx. Each kidney has got about 8 minor calyces and 2 to 3 major calyces. From minor calyces, urine passes through major calyces, which open into the pelvis of the ureter. Pelvis is the expanded portion of ureter present in the renal sinus. From renal pelvis, urine passes through remaining portion of the ureter and reaches urinary bladder. Hope you have understood the video. Like, subscribe and press the bell button for more videos. Thank you. Thanks for watching.